Hello and welcome. Tardigrades have become increasingly popular with the general public in recent years, not least because of their seemingly extraordinary survival skills. In general, we simply state these capabilities and leave it at that, keeping the mystery alive and leaving room for interpretation. In this video, it's a little different. The survival performances will be detailed but also explained, and you'll see that they're impressive, but not all that extraordinary when you know their secret. It's a subject that requires a mastery of various notions of chemistry, biochemistry and of course biology, to be understood in its entirety, but I'm going to simplify all these notions, and by the end of this video tardigrade resistance will no longer hold any secrets for you. All the concepts discussed are sourced. In the description of this video, you'll find a link to a file that lists all these sources, as well as further explanations if you'd like to know more about some of the concepts covered here, as most of them are very succinct. This video is divided into 6 main chapters. First of all, a few notions and contexts to try and define a minimum what life is and what we mean by living, followed by some basic notions about tardigrades. Then we'll talk about radiation resistance, but more importantly, the reason for this resistance, and we'll do the same with the other resistances possessed by tardigrades. Then, in the last 2 chapters, we'll put these facts into context to understand how these faculties originated. What is living? Well, the answer is actually very difficult to give. If you ask several biologists, it's likely that each of them will give you a different answer, because there's no real international consensus on this question. On the other hand, we can give some criteria that are similar to the living, ways of recognizing it. Living beings have a metabolism, i.e. they produce and consume energy to maintain themselves in a state of biochemical functioning. They also have a tendency to grow, i.e. they increase both in cell size and in cell number, in the case of multicellular organisms. Another common criterion is reproduction. Then we have a criterion that can be considered universal. In any living structure, we'll find a highly complex molecular structure. There's also homeostasis, which is the act of regulating one's internal environment according to external conditions. And finally, response to stimuli. The simple fact of shivering in response to cold, for example, is a response to a stimulus. These are possible criteria, but under certain conditions, living beings may no longer meet one of them. Viruses are generally not considered to be living organisms, as they don't have their own metabolism, but use that of their hosts. On the other hand, they reproduce and have a complex organization too, but we don't define them as living beings. It can also happen that living beings find themselves temporarily with a metabolism whose activity is undetectable, i.e. almost completely stopped. In this case, growth, reproduction and homeostasis are also halted. The response to stimuli may still exist, and it is by means of rather unexpected stimuli that biological activity would resume if the stoppage were temporary. This leaves complex organization as the only sure bet for detecting a living being, but it is not in itself sufficient, as there are other complex structures besides that of living organisms. It's a notion to keep in mind, especially for everything to do with exobiology, as we could encounter dormant life forms without realizing they're actually alive. In short, the living is a rather vague notion that we have great difficulty in defining. The boundary between the living and the inert is very diffuse, and cannot be seen as a clear line separating one from the other, especially as dormant life forms can blur the lines. To take this a step further, we can categorize the life forms we know according to their ability to slow down their metabolism. Although there are variations depending on the individual and physical activity, we'll assume that human beings are generally unable to slow down their metabolism. We will therefore say that their metabolic rate is 100% in all circumstances. This is not the case for overwintering animals, for example, and even more so for hibernating animals, where the slowdown is even more advanced. Among the most efficient in this respect are animals capable of freezing their metabolism and thus slowing it down considerably, and those that can literally stop it, or at least lower it to levels where it is undetectable, as in the case of rotifers, but also the tardigrades that interest us. Life is therefore sometimes very relative in terms of time. Where there's life, there's also environment. Here you can see a diagram of water phase transitions, and it's generally accepted that life evolves and thrives in environments where liquid water exists. However, there are 2 essential parameters for liquid water to exist: temperature and pressure. We're used to saying that water freezes at 0 degrees Celsius and boils at 100 degrees Celsius, but this is only true in our atmosphere at sea level, where the pressure is 1 bar. In the high mountains, however, this is no longer entirely true. This is one of the reasons why we can't really find stable liquid water on Mars, because at 6 millibars, the pressure of the Martian atmosphere is much lower than ours. Ice therefore sublimates directly into steam if the temperature rises. We therefore consider that habitable environments, as far as we can imagine, are necessarily included in this range of temperatures and pressures where liquid water can exist. At first glance, we don't expect to find life forms that can withstand conditions outside this zone. However, life adapts to a variety of environments, and we can distinguish two types of life forms. On the one hand, those we'll call classic, and on the other, extremophiles. This is mainly from a human point of view, as the classics are simply life forms that are adapted to habitats close to our own. Habitats that respect a range of conditions considered temperate. Pressure, for example, too low and you can't breathe, but too high and you'll be compressed and die. The same applies to temperature. Too cold or too hot and the organism will no longer be able to regulate its own fixed temperature to counteract the difference with the outside world. Humidity, gas levels in the breathing mixture, and pH, particularly for aquatic life forms, must not stray from a fairly narrow range either. These conditions are essential for survival. For extremophiles, it's the same thing. They need precise conditions to survive, but they're not the same as ours. From our point of view, they are extreme. There are extremophiles that live in acid, others at high temperatures, in salt, at high pressure, and so on. There are also polyextremophiles, which combine several of the extreme conditions necessary for survival. This is particularly true of life forms that colonize the black smokers at the bottom of the oceans, where the pressure is very high and the temperature high. If you put these extremophiles in our environment, you kill them, because they're adapted to an environment that's not ours. So they're no more resistant than the classics, they're simply adapted to a different environment. To these we can add a third category, extremotolerance. An extremotolerant is an organism that will live and thrive in a classic environment, but will withstand exposure to an extreme environment without being killed, notably because it will bypass this change of environment by slowing down its metabolism to circumvent the loss of conditions favorable to its survival. Our extremotolerant model, the tardigrade, is a phylum made up of numerous species. 1,300 have been recorded to date, but it is estimated that there are around 2,600 on the planet. It is also known as a water bear, because of its appearance. It's a microscopic animal, averaging 0.5 millimeters in length. It was first observed in 1773, in what is now Germany, thanks in part to the recent availability of microscopes. The presence of tardigrades is generalized to all habitats where liquid water exists at least periodically, which includes practically the entire planet. Note that this is a purely aquatic animal. It breathes oxygen dissolved in water, but cannot breathe oxygen from the air. And that's going to be important. The tardigrade is a fairly ancient phylum, and the history of our planet's evolution is summarized here to scale. First we have the Precambrian, which stretches from 4.6 billion years ago to 540 million years ago, when the Cambrian period began. This period is marked by what's known as the Cambrian explosion, simply because during the Precambrian, diversification was rather slow, whereas during the Cambrian, many new complex life forms emerged relatively rapidly. Biodiversity therefore exploded over several million years during this Cambrian period. And it was precisely during this period that the tardigrade phylum also appeared. We can therefore roughly say that tardigrades have existed as a phylum for 500 million years, but note that they are not the tardigrades of today. They have evolved and the species that existed then have long since disappeared. Today's tardigrades are, however, their descendants. Tardigrades are indeed complex animals. In the current classification of life forms, they belong to the eukaryotes, so their cells have nuclei and compartments, and many other typical features. As metazoan animals, they are multicellular and feed on and assimilate nutritious organic matter. They are bilaterians, their anatomical organization being symmetrical between right and left. Nephrozoans, because they have real digestion and excretion systems, just like humans, who are also nephrozoans. However, tardigrades are protostomans, unlike humans, because criteria are mainly embryological. Ecdysozoa, for morphological and reproductive criteria. Panarthropods, because they have a body divided into segments, appendages and a typical nervous system. Like arthropods, tardigrades are closely related to spiders and scorpions, the best-known arthropods. In fact, tardigrades seem to have followed an evolutionary path oriented towards miniaturization. Just like spiders, scorpions and other closely related animals, which have also reduced in size over the course of evolution. This is in contrast to certain other well-known evolutions. Non-avian dinosaurs, for example, evolved from small reptiles, eventually reaching gigantic sizes and masses of several tons. But they disappeared. So miniaturization may well be a better evolutionary solution. As we've said, tardigrades are aquatic and, like everything else living during the Cambrian period, they were originally oceanic only. It was only later that some tardigrades migrated to land. At present, there are purely oceanic tardigrades and others specialized in life on land, but still in environments with minimum levels of humidity. This excludes hot hyperarid deserts. They are therefore found in all continental environments where there is water, rivers, lakes, ponds, etc. But more surprisingly for an aquatic animal, they are also found in forests, meadows, mountains and even Antarctica. What we often forget to mention is that oceanic tardigrades don't have any truly spectacular abilities. The ones that will really push back the limits of survival are the terrestrial tardigrades, those that live in environments where the presence of water is not continuous, and we're going to see that there may be a connection. One of the most popular capabilities is resistance to radiation. To put this into perspective, let's compare it with the theoretical resistance of human beings. Here, with gamma rays. If you expose a group of humans to 5 grays of radiation, half of them will statistically die very quickly as a result of the exposure. 5 grays is therefore the median mortality dose for human beings. But for tardigrades of the species Milnesium tardigradum, which were tested in a study, groups of tardigrades saw their median mortality dose appear at 5,000 grays, 1,000 times the human dose. In other words, tardigrades of this species are 1,000 times more resistant than humans. Note that the gray is a unit, which means that you are subjected to radiation which sends you energy, expressed in joules, but taking into account your mass. A human being's mass is generally in excess of 50 kilograms, but a tardigrade's mass is tiny by comparison, a few micrograms at most. Proportionality must therefore be taken into account. To calculate very roughly, if a human and a tardigrade were both exposed to the same source of radiation, 200 microjoules would be enough to expose a tardigrade weighing 40 micrograms to 5,000 grays, but 500 J would be needed to expose a human weighing 100 kilograms to just 5 grays. That said, the tardigrade has a much smaller volume, which limits the radiation reaching it by the same proportion. Gray should not be confused with sievert, which measures the radiation you actually absorb, not that which is emitted. So, in any case, this is a very impressive result, all the more so as it is confirmed with other types of radiation. I encourage you to read the study, the link to which is available in the drive description. But what can we conclude? Is the tardigrade invincible? Not really. It's a median dose of mortality, which means that half the group doesn't survive, and the survivors themselves don't get off scot-free, with physical damage, infertility and reduced life expectancy. So it's very resistant, no doubt, but not invincible. This resistance is a fact, but how can we explain it? There are many hypotheses on this subject, particularly on YouTube and social networks, and at first sight attractive scenarios are quite widespread. Most of them point to an extraterrestrial origin and sometimes to exposure to radiation here on Earth, which would explain why he's adapted to resist it. There are other, less intuitive explanations, but first let's take a look at why attractive scenarios have some weaknesses. The first problem with invoking an extraterrestrial origin is the very makeup of tardigrade cells. They are animal eukaryotes, which means, among other things, that they have mitochondria, just like us, and that they breathe oxygen. What's more, they are aquatic and their appearance dates back 500 million years. They therefore necessarily appeared in an environment containing liquid water in which oxygen is dissolved, since they are adapted to it. This was the environment on Earth 500 million years ago, and it still is today, but is there another such environment nearby? In the solar system, we have Mercury, but it's very close to the Sun, with no atmosphere, no water and even less oxygen. Venus, on the other hand, has an atmosphere, but it's over 400 degrees Celsius. The pressure is 90 times higher than on Earth, there's no trace of water or oxygen, and this has probably been the case for well over 500 million years. Mars is a less hellish planet, but as the pressure is extremely low, there's no liquid water on its surface, no oxygen, and even if there were liquid water deep underground, there would be no oxygen production because there's no photosynthesis. For well over 500 million years here too. As for the moons orbiting Jupiter and Saturn, some have subterranean oceans, but oxygen production is ruled out. Even if microorganisms had developed there, photosynthesis would not occur, because under several kilometers of ice, darkness is total, and photosynthesis is a priori the only efficient way to produce oxygen in large quantities. It's possible to invoke an arrival from a distant solar system in an asteroid, but the distances are so gigantic that the journey would take millions of years. Tardigrade may be resistant, but it's unthinkable that such a long journey in the interstellar medium would be possible, as cosmic rays alone would have literally vaporized it. Two other arguments may suffice to refute this hypothesis. Tardigrades are not only eukaryotes, they also share many genetic similarities with the rest of biodiversity, as we have seen. This means that if tardigrades have an extraterrestrial origin, then so does all biodiversity. The final point is that oceanic tardigrades don't have these radiation-resistant abilities, they only appeared later, when they colonized dry land. The scenario of radiation exposure of terrestrial tardigrades also has some weaknesses. Firstly, there's no trace of an ancient radioactive episode. The ozone layer was already formed in the Cambrian, which limited UV radiation, and the magnetosphere that protects the Earth from a large proportion of solar and cosmic radiation has existed since the Earth's formation, so there's no natural source that could have pushed living organisms to adapt to high ambient radiation. Localized radioactivity is implausible, given the worldwide distribution of resistant species. What's more, terrestrial tardigrades aren't the only radiation-tolerant life forms. All over the planet, other resistant life forms can be found, such as scorpions, or the extremophile Deinococcus radiodurans, which outclasses tardigrades in terms of radiation resistance. Many life forms are not resistant to this type of stress, so it would be difficult to explain their survival if radiation had permeated the environment in the distant past. Obviously, the scenario explaining this adaptation isn't the most intuitive. It requires more thought, and we need to understand the mechanism behind this faculty in order to draw more verifiable conclusions. The question is, why are terrestrial tardigrades resistant to radiation? Is this really their objective? Not quite, in fact. They are in fact resistant to the consequences of radiation, and therein lies the subtlety. In fact, the consequences of radiation on living matter are the same as those produced by breathing and the use of oxygen, the possible presence of toxic substances and even dehydration. These consequences can be summed up as oxidative stress, i.e. the appearance of a high concentration of free radicals within the body's cells. Radiation resistance is therefore only an indirect capacity, the bonus effect of a much more general mechanism. To try and understand this a little better, we're going to take a look at some basic chemistry. Chemists won't be happy, as these are very simplified representations of atoms, and electrons behave in a much more complex way in reality, but a link is also available in the sources if you want to understand the organization of atoms in more detail. An atom is composed of a nucleus, itself made up of protons and neutrons. Around this nucleus are electrons in different layers, in this case a small atom, lithium, for ease of illustration. When it receives energy, which may simply be heat, it will become excited in response to this energy gain. But if the energy level received is too high, with a high energy photon coming into contact with it, gamma radiation, for example, the electron on the outer layer will be ejected from the atom. The atom is said to be ionized, and the ejected electron becomes free. These free electrons are particularly harmful in the presence of oxygen, as they modify its behavior and reactivity. To illustrate very simply what happens in an organism, we have oxygen molecules. Here we represent 2, but imagine multiplying that by billions of molecules. The free electrons generated by the ionizing disturbances will interact with these 2 oxygen molecules to form 2 molecules called superoxide anions, a more reactive species than oxygen. In an atom or molecule, electrons are naturally paired up. But these superoxide anions have a new electron which is therefore alone, making the molecule more reactive as the electron seeks to pair up with another electron. In a cell, many enzymes exist, including superoxide dismutase, which takes charge of these superoxide anions and neutralizes them by mixing them with two protons, or hydrogen nuclei, found abundantly in the cell. This will produce oxygen as before, but also hydrogen peroxide. This hydrogen peroxide will react with another free electron to split into two smaller molecules. The hydroxide ion, which isn't particularly problematic, but the second molecule is far more annoying, the hydroxyl radical. And this radical is the most reactive of all. This hydroxyl radical, represented more simply here as A1, is missing an electron to reach equilibrium, so it steals an electron from its immediate environment, in this case from molecule B1. The radical thus reverts to a common hydroxide ion, but in the meantime it's the molecule B that has had its electron stolen that becomes the new radical, since it's no longer at equilibrium. As its structure has changed as a result of the missing electron, the denaturation is irreversible. Not only will it no longer be able to perform its normal functions, but it will also steal an electron of its own to return to a state of equilibrium. It's a chain reaction in which each denatured molecule denatures another. This denaturation wave can affect proteins, lipids and, even more problematically, DNA. Molecules called antioxidants neutralize these hydroxyl radicals by giving them the missing electron, but without themselves becoming radicals. They remain stable even when giving or gaining electrons. But this system has its limits. If the reactive species are too numerous, as is the case with strong irradiation, only a small proportion will be neutralized by the antioxidants. To protect themselves from this very harmful phenomenon, terrestrial tardigrades have antioxidants, like all other species that live in contact with or use oxygen. They also have a highly efficient DNA repair system, replacing damaged areas as they are attacked by hydroxyl radicals. This system also exists in most life forms, but seems to be even more effective in tardigrades. But this is not the most effective defense system. Tardigrade DNA is in fact covered by a shield that prevents hydroxyl radicals from damaging it. It's this trick that makes tardigrades so much more resistant than others to oxidative stress, and therefore to radiation. This shield on DNA is formed by an accumulation of very specific proteins, which were discovered in 2016 and named DSUP, for damage suppressor. Take a cell unprotected by this system. When it undergoes oxidative stress and a massive attack of hydroxyl radicals, most of the cell's components will be damaged: lipids, proteins and, of course, DNA. As a result of this oxidative storm, the cell will have suffered a great deal of damage, but worse still, its DNA will have been totally destroyed, and there will be no possibility of repair. The cell will be dead. In contrast, a tardigrade cell protected by the DSUP shield will also suffer lipid and protein damage, but its DNA will not be affected. The cell will therefore still have the coding genes needed for self-repair, and will not die. It is this principle that enables tardigrades to resist radiation and any other source of oxidative stress. We may well ask what the point of this protection is, since it is not in fact designed to protect against radiation at all, but rather against other sources of oxidative stress. The main phenomenon against which it protects itself is actually dehydration and oxygen deprivation, but also its return during rehydration. These processes are major generators of oxidative stress. We saw at the beginning that tardigrades could stop their metabolism to protect themselves from an extreme environment, but for these aquatic animals, the absence of water is an extreme environment. They therefore practice what is known as cryptobiosis. Cryptobiosis is a state of stasis in which profound changes occur. The main trigger for cryptobiosis is anhydrobiosis, i.e. loss of body water. When tardigrade is 100% hydrated, its metabolic activity is at a maximum. When it becomes dehydrated because its environment dries out, it will gradually go into a state where it curls up on itself and its 97% body water has disappeared. Its metabolic activity drops to 0.01% of its active state, or 1/10,000, making it undetectable. When its environment rehydrates, the process reverses and the tardigrade returns to a normal level of hydration and metabolic activity. This is why tardigrades are found in environments such as forests and mountains, where they normally live during wet periods and enter a state of stasis during dry periods. Terrestrial tardigrades are not the only ones to possess this ability. There are other species that use the same survival technique, as shown here in two examples. But there are many others. Cryptobiosis can also be triggered by stimuli other than dehydration, such as cold, which also induces stasis through cryobiosis. The strong presence of water-soluble molecules such as salts, sugars or toxic compounds can also trigger stasis by osmobiosis or chemobiosis. Or a lack of oxygen in the water surrounding the tardigrade, known as anoxybiosis, but little studied. The technique can be summed up simply as temporarily ceasing to live in order, ironically, to survive. At present, we don't know how long cryptobiosis can actually be maintained. The current record is held by a tardigrade that spent 30 years in cryptobiosis in Antarctica. But there's nothing to say that much longer times aren't possible. It's hard to say which form of primordial cryptobiosis was the first to be used, but there's one very common environment where all this may have emerged: shorelines. With the tides, several changes can combine: salt levels, oxygen levels and, of course, dehydration when the water is completely withdrawn. So, perhaps it's all part of a whole. Cryptobiosis does have its weaknesses, however, and the survival rate of tardigrades practicing it is never really 100%. A 1989 experiment demonstrated this in a very common species. A group of tardigrades is rapidly dehydrated. The tardigrades go into cryptobiosis, then the group is rehydrated. None survived. Another group was dehydrated, but this time more slowly, and the tardigrades also went into cryptobiosis. The group was then rehydrated, and this time 88% of the tardigrades survived, and the researchers launched a new cycle where this time they rapidly dehydrated the same tardigrades that had survived, and by the time of the second rehydration 60% of the tardigrades had survived. This experiment teaches us two things. The first is that tardigrades need a certain amount of time to adapt to dehydration, as if they were putting something in place to prepare for it, and this something can be reused for some time, since they survive sudden dehydration after an initial progressive dehydration. The second is that some tardigrades never wake up from their cryptobiosis. A 10% loss may seem high, but a 90% survival rate is more than enough to gradually colonize all habitats and stay there. During this cryptobiosis, some astonishing skills emerge, the famous superpowers. They suddenly begin to resist temperatures far more extreme than those they withstand in their active state, ranging from -273 degrees Celsius, i.e. practically absolute zero, which is the minimum temperature where there is a total absence of heat, up to 150 degrees Celsius. This means they can withstand a temperature range of 420 degrees between minimum and maximum. For pressure, the second decisive environmental parameter for life, the resistance is even more impressive, since they can survive in a total vacuum, i.e. 0 bars, the conditions of interplanetary space, but also at pressure levels that don't exist on Earth, at least in the biosphere. To find the staggering pressure of 75,000 bars, they have to descend deep into the Earth's crust or generate it with technology. This ability to survive in harsh environments has aroused great interest in the scientific community, and several test missions into space have been launched. Here are two examples, and each time, a portion of the tardigrades have survived these conditions. Now the question is, how do they get into a state of cryptobiosis and come out alive? The culprit is once again a protein-based mechanism. Other cryptobiotic life forms use sugars such as trehalose to protect their organisms, but in tardigrades they were very rarely present, if at all. Instead, 4 types of protein were identified between 2012 and 2015, each with a protective role in a specific part of the cell or in the space between cells. Combating oxidative stress is a prerequisite for the success of cryptobiosis, but it's not enough. Water and life are intimately linked, even on a structural level. A loss of water inevitably causes cell denaturation, unless it's replaced, and that's precisely what we're going to see. Proteins and lipids are fundamental components of life. Proteins are assemblies of basic building blocks called amino acids, and among these amino acids, of which there are 20 in living organisms, some like water, i.e. they interact with it, and are therefore hydrophilic, while others don't like water, i.e. they don't interact with it, and are therefore hydrophobic. In simple terms, most proteins are made up of predominantly hydrophilic or predominantly hydrophobic moieties, arranged one after the other along a long chain. Classically, hydrophobic zones are hidden inside folded proteins so as not to come into contact with water, while hydrophilic zones move outwards, in contact with water. It is this configuration that actively contributes to the overall shape of the protein, known as conformation. Many factors come into play, disulfide bridges, ionic interactions, etc., but the idea is to highlight the decisive role of water, which is why we've set aside these other parameters. If the protein were to be dehydrated, one of the consequences would be unfolding. The protein would lose its conformation. And when the protein is rehydrated, it's unlikely to return to exactly the same conformation as it had originally. And that's a problem because the protein's function depends on its conformation, so this protein has become unusable. Imagine this happening to billions of proteins within cells, in which case metabolism is totally destroyed because reactions can no longer take place. Another form of protein denaturation is aggregation. Proteins bind to each other because water is a solvent. Proteins are dissolved in this water, and hydrophilic groups form bonds with it. If the water disappears, the proteins will start to form bonds with each other rather than with the water. Even with rehydration, it will no longer be possible to resolubilize them. And aggregated proteins also lose their function. Lipids, another fundamental component of life, operate on a similar principle. The basic unit of cell membranes, even if there are other players such as cholesterol, is the phospholipid. A phospholipid has two regions, one hydrophilic and the other hydrophobic. The basic building blocks that are these phospholipids are arranged in such a way as to form a double layer, where the hydrophobic parts come together and the hydrophilic parts interact with water. And once again, the water present in the membranes has an impact on the very structure that the phospholipids will adopt. If the membrane is dehydrated, it will become disordered and the phospholipids will change position. On rehydration, the structure of the membrane will never be exactly the same as it was before the temporary disappearance of water, and leaks will appear. Leakage is fatal to cells. A leaky cell is said to be in necrosis. It's for all these reasons that water is absolutely essential to life, and that living organisms have no apparent vocation to dehydrate and then rehydrate. As for the tardigrade proteins, there's something a little unusual about them. They don't organize themselves in the same way as conventional proteins, they're called disordered. When they're immersed in water, they move in all directions without ever managing to find a stable state, because the position of hydrophilic and hydrophobic groups on the chain makes this physically impossible. But where it gets interesting is when the water disappears, because the proteins will be able to stabilize and adopt a conformation, called an alpha amphiphilic helix, where they will have a hydrophobic side and a hydrophilic side. It is this conformation that enables them to replace water to prevent the denaturation of membranes and proteins. For the first type of protein, CAUS, which is found in the cell cytoplasm, when the cell is hydrated, these proteins have no significant impact. When the water disappears, unprotected cells see their proteins denatured, whereas in cells where CAUS are present, a mesh forms, and the CAUS proteins form a kind of highly viscous gel that appears and traps the proteins and even a small amount of water, so that they can neither unfold nor aggregate with each other. After rehydration, the water is stronger than the proteins, forcing them back into a disordered state, while the same water resumes its natural position around the protected proteins. Metabolism therefore suffers no significant damage. In the unprotected cell, metabolism is destroyed. In the case of the SARS protein, its action takes place outside the cell. It bathes in the extracellular environment and has no impact for the moment. Unlike the others, it is not composed of amphiphilic alpha helices but of beta sheets, another form of protein organization. When the extracellular environment becomes dehydrated, these proteins form a structure which prevents the cells from denaturing each other. Not only do they fuse together, they also prevent damage to plasma membranes. During rehydration, unprotected cells are necrotic and therefore destroyed, while cells protected by SARS are not damaged. The other two types of protein have a similar structure to CAUS, but essentially protect the membranes inside the cell, and in particular those of the mitochondria, which are the cell's energy powerhouses, supplying most of the ATP, the living world's energy source. When dehydration takes place in this zone, the MAS proteins will integrate between the phospholipids, as they have an amphiphilic structure. Their hydrophobic side will therefore be positioned towards the hydrophobic part of the membranes, and the hydrophilic side towards the hydrophilic exterior of the phospholipids. This will prevent the phospholipids from moving, so the membranes won't denature. When the water returns, the proteins destabilize and make way for the water, restoring the membrane's fluidity without damage. Membranes not protected by this system, on the other hand, will be denatured. To sum up this preservation mechanism, each type of protein provides protection in a different part of the cell, enabling it to survive dehydration, which is theoretically lethal to living organisms. DSUP reduces the oxidative consequences of this almost total dehydration by protecting the DNA. CAUS essentially protects the metabolic activity of the cytoplasm. SAS protects the exterior, and the other two proteins protect the integrity of the cell's energy plants. Let's see if there's a link between superpowers and resistance to temperature and pressure. So yes, there is a link. In the case of heat, the enzymes that make metabolism work only within a precise temperature range. For us, for example, cells must never stray too far from 37 degrees Celsius, otherwise metabolism stops, which is why your body constantly regulates its temperature. Tardigrades, on the other hand, prefer lower temperatures, around 20 degrees Celsius. Exposing it to heat when it's active will be fatal. But in cryptobiosis, metabolism is totally shut down, so exposure to high temperatures has no impact. Up to a certain limit, because even disordered proteins are denatured at high temperatures, including the protective proteins he uses. What's more, at very high temperatures it will simply burn. In the case of cold, a hydrated living cell will see its water turn to ice and form crystals, and crystals tend to denature cells because they are sharp. There's also the problem of metabolic activity, which requires a temperature interval from which to exit. What's more, ice takes up more space, so the cell tends to burst. In cryptobiosis, on the other hand, water levels are infinitesimal, so the problem no longer arises. There will be no impact. At the pressure level, in a state of cryptobiosis, these are no longer fragile cells, thanks to the protein structures that have formed to protect against dehydration. A compressed hydrated cell will burst under pressure, whereas a dehydrated cell with the consistency of a viscous gel is much more malleable and will at most slightly deform. The cell then resumes its initial shape, without being damaged, unlike a hydrated cell. To make an analogy, we could compare it to the fable of the oak and the reed. The structure formed by the protective proteins is rigid but not too rigid, just flexible enough to deform without breaking, like the reed, which is why it resists gigantic pressures when the tardigrade is in cryptobiosis. We saw earlier that cryptobiosis doesn't work if tardigrades are dehydrated too quickly. This is because the preservation proteins are not present in large quantities and continuously in the cells, except for DSUP, which is continuously expressed. For the other proteins to be produced, the organism must first detect that environmental dehydration is taking place. This detection produces what is known as a signaling cascade. To illustrate, there are 5 stages, but there may be many more. Here we have our detector A, which activates and releases a molecule B, which in turn activates C, which in turn activates D, and so on down to the cell nucleus where the DNA is located. Compound E unlocks access to the genes that code for the production of resistance proteins. Enzymes known as polymerases will then be able to read these genes and produce RNAs, which can be seen as photocopies of assembly instructions encoded in DNA. These RNAs migrate to the cytoplasm to join elements called ribosomes. The ribosomes assemble chains of amino acids in a precise order communicated by the RNA, and this is how the desired proteins are obtained. This applies to all proteins in the body. Some proteins also contain a code that enables them to be taken up by the cell's transporters, for example to the mitochondria in the case of the MAS and LEAM proteins. This is how tardigrades react to dehydration and prepare for cryptobiosis. This takes time, and dehydration that is too abrupt prevents them from completing this process. So, we've seen how superpowers work, and why they're perfectly explainable, even if, of course, not everything has yet been elucidated in detail. Now let's take a look at how we got here, how these mechanisms came into being and under what circumstances, with a brief summary of the evolution of life on Earth to set the scene. I'll keep the dates very vague, however, as they may be refined as new discoveries are made. First of all, our Sun and planet were formed around 4.6 billion years ago. The ocean hasn't yet formed and the atmosphere contains no oxygen. The ocean was formed during the cooling of the Earth, and it is assumed that the first forms of life, very simple compounds, appeared around 4 billion years ago, but it's very difficult to date precisely, as there are no fossil traces left from this period. For the time being, there is no oxygen either in the ocean or in the atmosphere. Life is therefore anaerobic, not breathing, but already beginning to evolve. Sometime after this start, one life form in particular stood out from the rest, since it was the only one to leave descendants, including us tardigrades and everything else living on Earth. This is Luca, the last universal common ancestor of all life forms on Earth today. A decisive element in the evolutionary history of life on Earth was the appearance of photosynthesis, which changed everything. This process produces energy from water, carbon dioxide and sunlight. At the same time, it releases water and oxygen. This oxygen is an unused waste product in this ancient age, and will soon have consequences for other forms of life. At least 2.8 billion years ago, cyanobacteria, descendants of the first photosynthetic organisms, appeared. They pushed oxygen production to extreme levels, triggering the first great genocide in the history of life on Earth. By colonizing the ocean and poisoning it with oxygen, they caused the disappearance of the majority of anaerobic life forms that were not adapted to the consequences of oxidative stress. Today's cyanobacteria occupy every environment on the planet. While most microorganisms succumb to toxic oxygen, some are lucky enough to evolve and develop respiration, a process that uses oxygen to produce energy. These are the first aerobic bacteria. They consume the oxygen released by the cyanobacteria and release carbon dioxide, but the imbalance is too great as the cyanobacteria reign supreme and oceanic and atmospheric carbon dioxide levels begin to fall as they consume it. Between -2.4 and -2.1 billion years ago, what had to happen happened, and carbon dioxide levels having fallen too far, the greenhouse effect was no longer sufficient to maintain a mild average temperature. This led to the Huronian glaciation, which lasted 300 million years, with a thin band of liquid water probably persisting at the equator. This is known as the Great Oxidation Event. Oxygen is highly reactive compared with carbon dioxide, and after oxidizing the metals present in the ocean, saturating the ocean itself, it spreads into the atmosphere, oxidizes the surface metals and then begins to accumulate in the air. This also forms the ozone layer at high altitude. At the end of this great oxidation, cyanobacteria won't stop there, but an event will re-establish good temperature conditions. After 300 million years of ice age when everything could have stopped there, massive eruptions release large quantities of carbon dioxide and thus re-establish an acceptable greenhouse effect, which melts the ice and reshapes the ocean surface. Between this period and the Cambrian, numerous revolutions took place, but the list is by no means exhaustive. First of all, the appearance of multicellular life, where cells began to cooperate to form a single organism. Aerobic bacteria will be captured by more massive cells that can't breathe and are therefore less profitable in energy terms. But digestion doesn't take place, and a synergy is created. The bacteria produce energy by breathing in oxygen, while the massive cell provides protection and nutrients. This association, resulting from endosymbiosis, proved to be extremely efficient, and is at the origin of the animal kingdom, among others. The mitochondria of our cells are therefore the descendants of ancient aerobic bacteria. A second phase of this endosymbiosis process also occurs, but this time with a cyanobacterium. Endosymbiosis is even more efficient, as the resulting cell only needs water, light, carbon dioxide and oxygen, which it also produces. It doesn't need any organic matter, as cyanobacteria produce it from carbon dioxide and water. These were the first plant cells. The descendants of captured cyanobacteria are called chloroplasts. The emergence of sexual life was also a key phase that led to an increase in biodiversity and the appearance of new, complex species. Between -540 and -480 million years ago, we are finally in the Cambrian, where complex life forms are now rapidly diversifying, and in which we find the ancestor of tardigrades. Around 480 million years ago, for the first time since the appearance of life, an animal walked on land. The appearance of the ozone layer millions of years earlier made the UV index acceptable, and some animals developed primitive lungs compatible with breathing atmospheric oxygen. This is a different evolutionary path from that followed by tardigrades. You'd think that things would be running pretty smoothly by now, but not at all. The Earth went through several periods of intense upheaval, forcing life forms to evolve and develop new skills in order to survive. 440 million years ago, a new ice age wiped out 85% of all living species in a relatively short space of time, but still counted in millions of years. Once the crisis had passed, life rediversified, but was once again confronted with a mass extinction around 374 million years ago, this time 75% of species disappearing as a result of a drop in oxygen levels. If you're looking for a definition of the apocalypse, then the Permian-Triassic extinction event of 252 million years ago might fit the bill perfectly. The volcanoes that gave rise to the Siberian traps erupted and the carbon dioxide released caused such intense acidification and heat that 96% of living species disappeared. 201 million years ago, volcanoes once again disrupted biodiversity, but this time they were located in what is known as the Central Atlantic Magma Province. This new mass extinction eliminated 80% of species, but paved the way for the reign of the dinosaurs. Finally, the best known of the 5 mass extinctions marks the end of the reign of the non-avian dinosaurs, with which 75% of species disappeared. Once again, volcanoes were partly responsible for this extinction, but the fall of an asteroid greatly aggravated the already difficult survival conditions caused by intense volcanism. In the meantime, tardigrades are doing rather well, although species diversity was undoubtedly reduced at the time of the mass extinctions. It's important to note that life is constantly evolving, and that from the Cambrian to the present day, the number of complex species is probably in the billions, without bacteria, because their diversity is so enormous that it's difficult to estimate. The most optimistic estimates put the number of complex species in existence today at around 20 million. In other words, there's not much left of the diversity of the ages. In today's diversity, we can only see what has been able to evolve to circumvent the various pressures that have shaken the environment since that time, and which have therefore been able to leave descendants. So all this is a way of putting things into context and showing some of the decisive phases that life has had to face in order to continue developing. Now the question is, how does life evolve? To understand the evolutionary process, we need to look at DNA. When cells divide, they copy, or duplicate, their DNA, but it's very rare for the copy to be exactly the same as the original, due to the very physics and material imperfection of polymerization processes. And one of the possible errors, known as mutations, is gene duplication. It can happen that base pairs, whole genes or even entire chromosomal regions copy themselves 2 times instead of once. This is a relatively common mistake, and a fundamental driver of the evolution and diversification of life on Earth. But only if this mutation is passed on to descendants, so it's only of interest in germ cells. From a dividing mother cell, we obtain cell 1 and cell 2. Assuming we focus on a small region of 3 genes, we can see that cell 2 now contains twice the version of gene 2. Cell 2 will therefore have 2 genes that code twice for the same protein, shown here in red. So far, nothing extraordinary. Eventually, we may see a beneficial effect if this protein is produced more efficiently. In some cases, an excess of a certain type of protein can also have a pathological effect. But in absolute terms, we will consider that in this general case there is no really significant impact. In the meantime, the cell continues to divide and new mutations appear. In one of its descendants, we imagine that another possible mutation, of which there are 4, appears in one of the 2 twin genes. Here, we'll imagine an insertion. This is when extra bases are added during duplication. The result is no longer 2 identical genes, but one that has retained its original function, and the appearance of a distinct new gene that gives a new function to a new protein. If this protein is harmless or useful, the gene will be passed on, as the individual will survive and reproduce. If it is harmful, the individual will not survive. If we repeat this scenario thousands or millions of times, we end up with thousands of genes present in the genomes of different species, which is precisely what we have today. To transpose this general principle to tardigrades, the most likely scenario is that tardigrade ancestors possessed an ancestral gene very common to dehydration-resistant species, a gene that codes for a protein involved in late embryogenesis processes, the Li protein. Found notably in drought-resistant plants, it is intrinsically disordered and quite similar to the protective proteins used by tardigrades. If we imagine that it was this type of gene that they originally possessed, which was copied and whose copies mutated independently to give rise to the gene families present in today's tardigrades, then their abilities become much more comprehensible. That was for the genetic side, but it's not enough for a mutation, which is totally random, to appear for it to be transmitted and end up becoming an integral part of the genomes of a new population. There's another factor that selects or rejects these mutations: natural selection. To illustrate this, imagine two populations of tardigrades of the same species, some with interesting mutations that make them slightly more resistant to dehydration than the others. One population wisely stays in the ocean, the other has unwisely ventured into a lagoon. Environmental changes occur and the consequence is a drop in sea level. Tardigrades remaining in the sea are not impacted, and therefore experience no selection pressure. On the other hand, the water in the lagoon will evaporate, exposing population 2 tardigrades to dehydration, so selection pressure is underway in this environment. An episode of rain rehydrates the lagoon and the tardigrades that survived the dehydration are able to come back to life, while those that were genetically too unprepared are dead. Those that were sufficiently resistant will reproduce again and pass on these abilities. Multiply this by thousands or millions of cycles and eventually, the population of tardigrades in this lagoon will consist solely of tardigrades that are highly efficient at resisting dehydration and rehydration. It's likely that this is how terrestrial tardigrades acquired all their superpowers. And as with all species, evolution continues. If you've followed this video so far, I'd like to thank you. The aim was really to show that a complete scientific investigation allows us to understand this subject, even without mastering all the biology disciplines used here. And that you should be wary of what you hear, particularly on the internet, about scientific subjects that at first glance seem inexplicable. If you'd like to delve deeper into certain subjects, each slide is numbered and in the description there's a link to a drive where the necessary sources are classified according to slide numbers. Thanks again for your attention.